This video is to help you to prepare for the cellular respiration lab, where you're going to be testing the effect of some kind of environmental variable on your choosing, so it's an inquiry lab, on the respiration rate. And so we're going to be taking a look at this equation for cellular respiration here. Cellular respiration is something that all living things are going to need to be able to do. Glucose and oxygen are required in the mitochondria, and from that, the products of this reaction are carbon dioxide and water. The useful end product, now this is kind of a biologist's explanation up here rather than a true chemist's chemical reaction. Notice it's not even balanced. But we are getting some useful energy, and that's the main end point on it. We'll talk more about that in class. So generally what we're going to be taking a look at is we're going to be taking a look at the amount of, in our particular experiment, we're going to be taking a look at the amount of oxygen that is consumed. And if we're looking at the amount of oxygen that is consumed, more oxygen consumed means more cellular respiration. All right, so in order to set this up, we're going to use some respirometers. So I'm going to slide this over here a little bit so you can see the respirometer diagram. Okay, so we're going to use some respirometers. This is what they really look like. There's a glass vial, stopper, and then a graduated, see the graduations, it's a graduated, it's got lines on it and numbers, pipette attached. All right, so in the respirometer, one of the things that we're going to need to be able to do is we're going to have to take carbon dioxide out of the equation. Otherwise, as oxygen is consumed, carbon dioxide is produced, and you don't see any kind of a measurable change. So we need to put down in the bottom of each one of the respirometers that you're setting up, you're going to need to put down there a cotton ball. And you're going to have more than just 2 respirometers. You probably are going to have in the neighborhood of about 8 of them. All right, so in the cotton ball in the bottom, that's not just taking the carbon dioxide out, but what we're going to put into that cotton ball is going to. So in the cotton ball, you're going to put in 1 milliliter of potassium hydroxide. Potassium hydroxide is going to absorb that carbon dioxide, form a reaction, reacts with it, and it takes it out of the air and puts it into a solid form that basically is volume negligible for us. So that's going to be taking that out of the equation. Therefore, any changes in the volume of a bubble of gas that's enclosed in this respirometer are just due to oxygen consumption. All right, so we're going to have that in there. But the thing about potassium hydroxide is it's kind of a caustic substance. It's not a substance that we want our peas sitting right on top of. So besides a cotton ball in the bottom that you're putting the milliliter of potassium hydroxide, you're going to put a layer of rayon in between here. Very thin, just enough so that your peas aren't sitting on the potassium hydroxide. So this is rayon here. Okay, it's another kind of cotton ball type substance. Okay, and then we're going to need to put some peas in here. So I'm going to have some peas that have been germinating. I recommend putting about 15 peas in kind of a standard setup. So we'll put about 15 germinating. So they're growing, they're sprouting. Peas in a standard respirometer. So this would be kind of a typical experimental setup. So what's going to end up happening with this respirometer then is we're going to allow it to equilibrate. I'm going to give you guys some steps to start with, to work with when you're making a procedure. But then what you're ultimately going to do is you're going to sink it. You're going to put it in a tub of water. Okay, and this is kind of what it looks like. Okay, so initially, can we see it? Okay, so initially they're kind of sitting like this. You give them some time to equilibrate. But then this is filled with water, and you put it underwater. And so then what you have is you have a bubble of air that's inside of the respirometer that we can measure the size of that bubble using the graduated marks on there. That bubble with oxygen consumption should get smaller. Okay, so back up to the board. Okay, so we're kind of expecting this bubble of gas that's inside of here to get smaller. So you're kind of expecting in a standard setup that the line where the bubble is going to be moving toward the vial as time goes on. We're going to be measuring how fast does that happen. That's your rate. But the thing is, remember from physics class perhaps, or chemistry, the gas laws, PV equals nRT. So we have an enclosed volume of gas here. So there are some other things that can affect the size of that volume, the size of that bubble. One of those, so we've got PV equals nRT. Temperature is something that's we really need to make sure that we keep constant. And pressure changes, even little weather changes. This is a very, very sensitive setup. Any little waves in the container, any of this can influence the size of this bubble, and we just want to measure the amount of change that is due to cellular respiration alone. So we're going to need a control setup to compare it to, one where you have no respiration occurring. So what you're going to do is you're going to, instead of having germinating peas in a setup, in each water bath that you have running, you also are going to need to have a setup that has, instead of peas, it has beads. And you want them to be the equal volume of beads. Okay, so you're going to have to figure out what is the volume of your 15 peas. You're going to do that by displacement. Okay, so you're going to have a graduated cylinder, put 50 mils in it, pop in your beads, and see how much the water went up. So you'll know the volume of your beads, or your peas. And then you'll do the same thing for the same amount of volume, but we're going to put in there plastic beads instead. Okay, so these plastic beads, it's probably going to be about 15 there. I think they're roughly meant to be the size of a pea, but it depends upon your peas and things. So you're going to also have this setup that should not be doing any cellular respiration. So if there's any changes that happen in this container that have the beads only in it, then you know that you can subtract that off from the changes that are happening in the pea only respirometer. Okay, so you and your group are going to be thinking about and brainstorming about what environmental variables could influence the respiration rate, possibly in peas, but I suppose it's possible that you might want to consider other organisms as well. We could do anything you could fit into those vials. You could do different kinds of seeds. I don't want to lead you guys too much. So I want you to think about what you might want to do there. Certainly ask me if you have any questions about materials that are available to you, or whether that setup would work, whatever variable you're talking about. Try to keep it something that is relevant, though, okay, something that matters to the peas, and like plant germination or something that matters in the world. Okay, on the day of setup, when you come in on that Monday, it's going to be really important that you know exactly what you're doing. This is going to be a day where we are going to be hurry to get started so that we can collect as much data as possible. Warning you guys on this, you have to know what you're doing. So when you come in a group of 4, I would expect to have people that know what their jobs are. These are the roles that I need to be able to do. And as soon as you come in the room, get started, get it set up. First hour people, you might want to even come in 5 minutes early. All right, so there's going to be one person that should be in charge of setting up the vials, where they're putting the cotton balls in, they're putting the potassium hydroxide in, they're going to be putting the rayon in, and then just kind of making sure that everything gets assembled once the other people are finished with measuring the peas. I think it works best to have 2 people that are working on the volume of the peas, because there are going to be, like I said, about 8 vials that I want you guys to get set up, probably, depending upon your independent variable. And by the way, with your independent variable, you're only going to probably be able to do 2 different variations on it. So let's say you're changing temperature, you're either going to be able to do like a warm and a cold. You're not going to be able to do 3 different ones just because of the limitations with our setup. Okay. So we have 2 people that are working on what is the volume of these peas by displacement, and then figuring out the beads as well so that you can get that set up. Remember, you need a bead setup for each water bath that you have going. And then you should have, if you have a 4th person, have them be in charge of the water baths. The water baths, maintaining temperature in them is going to be really important. Like I said, PV equals nRT, and so they're going to have a thermometer in there. And if you're dealing with cold particularly, they need to make sure that that temperature stays constantly cold and that you're adding ice as needed. So water bath person, and then that person would be general help. If you have a group of 3, then this person probably is just going to be, the other people will be working on setting it up. So those are the tasks that I'm looking that you guys know. You know your group is, you know what you're going to be doing, so that when you come in on that Monday after Thanksgiving, that you are immediately getting started on this. And like I said, if you can get here a little bit early, first hour, that's even better.